Hi, my name is Dmitri. I'm 34 years old, and I just finished my PhD project here. I am a neuroscientist now, I guess. During my PhD, I started with focusing on how excitability of neurons and how they could change their intrinsic excitability. And then we tried to scale it up to the network, and along the way we found some very interesting things also about not only how neurons and networks can regulate themselves and balance themselves, but also how they can tune to certain frequencies which can affect the whole network actually. One of my predecessors, they found that pyramidal neurons were able to upregulate a certain current, the IH current, which is also found in the heart as a pacemaker. And they upregulated it with increased excitatory input. And what the upregulation of that current basically did is that it reduced the excitability of those neurons. And my PI basically got some money and a project available to further investigate it. But there was not a very clear specific conditions on forehand. So it was very scientific, basically like, hey, we want to know more about this and please go ahead and investigate. So we started with the idea then to investigate if other types of neurons also had the same property. And we focused on interneurons first because they are very opposite from the pyramidal neurons, of course, in functioning. And it was very cool that we found already very quickly that we already knew that they had the same current, but we found very quickly that they also upregulated it in the same way as pyramidal neurons did. Yeah, so I did all my research in vitro, and we basically used this setup for almost everything. And what you see here is the microscope. It's very nice that this microscope is automated. Basically, this microscope enabled me to do the double patches. I also tried it on another setup and it was a lot harder, and the failure rate was a lot higher. And also the manipulators here, what I really like about those is that they are very precise and they're automated, and you can set their positions. So it made it very easy to basically set a start position, also measure different things and settings, measure where we were compared relatively to each other. My book is about balancing the brain. It's a summary of all my research. And we started with the question on single cells, how they can balance themselves and how they can prevent that they become too active or not active enough. And then we scale up to a network level where we want to see how it affects a micro network. Of course, a network is more complex and the brain consists of many more cells. So we had to make a selection, and we chose a very basic and well-established microcircuit in the hippocampus. And there we found that actually changing this property also changes the excitability, but also the resonance frequencies of the network. And that's really interesting because that's linked to a lot of ways of how the brain is able to process information and store information. Then next we provided a new method, which is basically not new, but a technique we developed here which can enable every lab to manipulate neurons in their own lab using this equipment for less than $100. And they can basically mimic every type of channel or current which these neurons might have, and they can mimic it live and see what happens if you change it. And this is a technique which was there already for a long time, but it needed large dedicated systems. And I think it's so cool that we got to an age in technology now where, well, basically your smartphone can do it, and you have very cheap chips which can do the same, but they're so easy to modify to your needs. So yeah, if you have all the recording equipment and you are able to patch the cells, you can connect 8 or 9 of these systems in parallel if you want. That's such a powerful technique, and it will help us to learn so much more about the brain than just using the standard blockers or enhancers. Yes, we found a clear difference between pyramidal cells and OLM interneurons. It was already known that interneurons fire a lot faster than the pyramidal neurons, and they have to because they are the brakes of the brain, so they have to respond very quickly. But it was not that clear what drives these differences, and if you could theoretically, by inserting these properties into a pyramidal neuron, turn it into an interneuron-like firing-like cell. And it was very cool because we used a technique in which we developed in the previous chapter, and with that technique we could show that we were able to change some of the basic properties which make the OLM cells and put them into the pyramidal cells. And that was so cool because then you could see a clear difference in the firing pattern, and it might explain how these small differences or changes can produce these different cell types. And before I showed that some of these properties are very dynamic, so that also means that probably a lot of our cells in our brains can adapt very dynamically in their output and their function, and in that way keep the brain stable. But also it might relate to a lot of pathology, of course, because you can imagine that if you have this high adaptation and it fails somewhere, that probably a lot will go wrong, because that's probably the dynamic regulation of the brain. Scientifica supported me with all the equipment, of course, because without that I would not be able to do any of these recordings, but also because they were very generous and they gave me contribution in the printing of the book, and as a PhD student that's very welcome. Accessible for free on the website of the University of Amsterdam. They have a system where they index it. It's called DARE, and people can find it there for free. Some of the stuff in the book is not published yet, so there's an embargo on it, but it will be released as soon as it's published.